Real-time PCR or quantitative qPCR. This technique can be used to quantify amplification reaction as they occur in real time. They need a special thermal cycler that uses a laser to scan a beam of light through the top or the bottom of each PCR reaction, and each reaction contains either a dye containing a probe or a DNA binding dye that emits a fluorescence light when illuminated by laser. The light emitted by the dyes correlate with the amount of PCR product amplified. Then the light is captured by a detector and the amount of fluorescence is read by software. The readout is plotted and analyzed to quantify the number of PCR product produced per each cycle. There are two approaches. Either use a Tachman probe, and in a Tachman probe it's actually a short sequence of DNA which is complementary to a specific region of target DNA between forward and reverse primer. This Tachman probe contains two dyes, a reporter dye at the 5' end and the quencher dye at the 3' end. And the other one is cybergreens, which binds to, it's another type of dye, can bind to newly formed double-stranded DNA and add more, as more double-stranded DNA is copied with each round of qPCR, there are more DNA copies, so they are more cybergreen, which emit lights under fluorescence, which emit fluorescence under the UV. This diagram summarizes the steps of a qPCR. You can see that the primers, forward and reverse primer, anneal. Then you have another primer which have the reporter and the quencher at the 5' end and 3' end respectively. Soon after that, the polymerization starts from the forward primer and the reverse primer. Once the polymerization reaches a reporter molecule on the 5' of the probe, it will start, upon the excitation of UV, it will start to make fluorescence. And here you can see how it is read out on the computer software, so you can see how the curve looks like for high gene expression and low gene expression. In situ hybridization is a type of hybridization that uses a labeled complementary DNA, RNA or modified nucleic acid strand, like probe, to localize a specific DNA or RNA sequence in a portion or section of tissue in situ. Gene microarray. Gene microarrays enable researchers to study all the genes expressed in a tissue very fast. This gene chip, the microarray, is created with use of small glass microscopic slide. The single-stranded DNA molecule are spotted on a slide using an arrayer, which is a computer-controlled robotic arm, which fixes DNA or multiple copies of cDNAs at different spots on the slide, and which is recorded by a computer. The procedure actually extracts RNA from a tissue of interest and does a cDNA by reverse transcription from the messenger RNA, and you label with fluorescence dye. This labeled cDNA is incubated overnight on the array, where it hybridizes with different spots on the array that contain complementary sequence. This array can have more than 10,000 spots of DNA. The array is washed and scanned by laser that causes cDNA to hybridize to the array. The cDNA which already hybridized to the DNA which exists on the array will fluoresce under excitation. These fluorescent spots reveal which genes are regulated, and the intensity of fluorescence in this indicates a relative amount of gene expression. These figures summarize what's happening in the microarray. So we first extract RNA from the tissue sample, then you do reverse transcription number 2 to get the cDNA, then you incubate this cDNA molecule, you do it a single strand, melt this cDNA molecule, and then you get the array, which looks like the figure on the top right. You can see incubate this single-stranded cDNA with the array, which have another a lot of 10,000 spots of cDNAs. So once this cDNA have a complementary sequence on the array, it will bind, and once you excite light on it, the computer software will detect based on the amount of light. If it's a bright fluorescence, highly expressed gene is present. If it's no fluorescence, there is no expression of that gene. If it's moderate, this means low gene expression. Gene mutagenesis studies. It's used to study the structure and function of protein produced by a specific gene. Site-directed mutagenesis is used to create specific nucleotides of a clone gene contained in a vector. Genes is then expressed in cells, which result in translation of a mutated proteins. So why use this approach? The procedure lets researchers study effects of mutation on protein structure as well as provide information about which nucleotides are important for a specific function of a protein. RNAi. Naturally, these RNAis are naturally occurring mechanism for inhibiting gene expression. Its procedures, actually the double-stranded RNA can be bound by a dicer enzyme that cuts the double-stranded RNA into around 25 nucleotide fragments, and these are called small interfering RNA, siRNA. This siRNA can be bound to a RISC RNA-inducing silencing complex. This RISC unwinds, melt the double-stranded RNA, releasing single-stranded RNA that can bind to messenger RNA of your gene of interest. The binding of siRNA to this messenger RNA leads to the degradation of the messenger RNA by a silencer enzyme or blocks the translation by interfering with ribosome binding. Currently, there are many developing techniques to use RNAi to silence gene expression. This figure summarizes how RNAi works. So you have a double-stranded RNA with the aid of the dicer, and the dicer will break this long double-stranded RNA into short ones. Then the RISC, including the silencer, will melt this RNA into small fragments called siRNAs. This siRNA will bind complementary to the messenger RNA of interest. You want to knock down it or down-regulate it. So it will either degrade messenger RNA by silencer or inhibition of messenger RNA by interfering of the binding of the ribosome to the messenger RNA. The second topic is genomics and bioinformatics. Actually, it's a hot discipline in biotechnology. Genomics actually it's a cloning, sequencing, and analyzing entire genome by a process called shotgun sequencing or shotgun cloning. They use restriction enzyme to digest pieces of entire chromosomes. The procedure will lead to thousands of overlapping fragments called contigs, which are each sequence. So you have a lot of contigs, you sequence each of them. The computer program will align these sequences based on overlapping sequences pieces. Bioinformatics, actually it merges the molecular biology with computer technology. This interdisciplinary field applies computer science and information technology to promote an understanding of biological process. Its application actually used it as a database to store, share, and obtain the maximum amount of information related to gene structure, gene sequence, and expression, and the protein structure and function. The gene bank actually is a public database of DNA sequence and contains all the National Institute of Health collection of DNA sequences. Each entry has an accession number. Each entry of the genes of the sequence of the gene has an accession number that scientists use to refer back to the clone sequence. These are maintained by the National Center for Biotechnology Information, which is NCBI. Basically, actually, it's a PubMed research database that you use to search for any paper you want to read about research. NCBI is a gold mine for bioinformatics resources that creates a public access database and deploys computing tools for analyzing and sharing genomic data. You can see here how we do this sequencing. You can see that the genomic DNA can be cut by a number of enzymes. A lot of fragments will arise. This collection, we call it library or contigs. These contigs can be aligned used by computer database to determine the sequence of these pieces, and they align to know the unknown sequences. And here you can see how they overlap the sequence to see the complete sequence of a DNA of a specific region. They can use as well a DNA database searching tool, which is BLAST, Basic Local Alignment Search Tool. This BLAST actually they compare the sequence with other sequences in other organisms to see if it's a complete match or no match. So in this figure you can see the human OB gene is very similar to the mouse OB gene. The Human Genome Project. Starting in 1990 by the US Department of Energy, In April 14, 2003, the human genome have been completed, map. The international cooperation of efforts to identify all human genes and to sequence all the base pairs of human chromosomes, mostly done by 20 centers in 18 countries, as you can see. The Human Genome Project HGB designed to accomplish the following: analyze genetic variation among humans, that means SNPs identification, map and sequence the genome of model organisms, including bacteria, yeast, roundworms, fruit flies, mice, and others, develop new laboratory technology such as high-power automated sequencers and computing technology, as well as widely available database for genome information, which can be used to advance our analysis and understanding of gene structure and function. It is used as well to disseminate genome information among scientists and general public. They consider the ethical and legal and social issues that accompany the HGB and genetic research. So what we have learned from HGP: nearly 15% of genes do not yet have a function. The summary of the finding of the HGP project: 3.1 million base pair make up the human genome. The genome have approximately 99.9% are similar between individuals of all nationalities. There is a lot of SNPs or CNV copy variation number, such as the long dilation, insertion, duplication in the genome, and account for much of the genome diversity identified between humans. Less than 2% of the genome codes for the gene. The vast majority of our DNA is non-protein codon, and there is a lot of repetitive DNA sequence account for at least 50% of the non-coding DNA. The genome contains approximately 20,000 protein coding genes. Many human genes are capable of making more than one protein. That's because, of course, of the alternative splicing, allowing human cell to make at least 100,000 protein from only about 20,000 gene. Chromosome 1, which is the biggest chromosome, have the highest number of gene, and the chromosome Y, which is the smallest chromosome, have fewest gene. Many of the genes in a human genome show a high degree of sequence similarity to genes in other organisms. Thousands of human genes have been identified and mapped to their chromosomal location. The top figure is for knowledge. You can see on the x-axis the number of chromosomes, and you can see on the left y-axis the number of genes it contains. On the right y-axis you can see the number of base pairs it contains. The Human Gene Project have started another expression called omics. It makes a revolutionary expression, omics, in which in proteomics it is studying all protein in a cell. Metabolomics is studying proteins and enzymatic pathway involved in cell metabolism. Glycomics is studying carbohydrates of a cell. Transcriptomics is studying all genes transcribed in a cell. Metagenomics analysis of a genome of an organism in an environment. Pharmacogenomics customized medicine based on person's genetic profile for a particular condition. Nutrigenomics interaction between genes and diet. Comparative genomics, it is the sequencing of genome for a number of model organisms. Stone Age genomics, Genomics, is analyzing ancient DNA. This figure for knowledge is showing the proposed function of a number of human genes assigned to a functional category. And you can see from this figure, the blue area, that you have around 41.7% of the genes of unknown molecular function. This table as well is for knowledge, but it shows the comparison of some of the selected genomes. So you can have the organisms, the approximate size of genome, the number of genes that these organisms have, the approximate percentage of genes shared with human, and how we get this information through the internet. Thank you.